Welcome back to the University of Melbourne Biology series. This is episode 14a, and today we follow one of biology's most astonishing journeys, how a single cell becomes a whole organism. Hold that thought for a moment. You began life as one cell, a fertilized egg, and yet you are now built from a vast number of specialized cell types, each doing a particular job in a particular place. Nothing about that arrangement is random. Over the next 12 minutes we will trace that journey step by step. We will watch the early embryo take shape, ask how one cell can give rise to many different kinds of cell, look at the cells that keep the body running, and finish by stepping back to see the overall body plan of an animal. Let us begin at the very beginning. It starts with embryogenesis, the process by which a multicellular organism forms from a single-celled zygote. This is no chaotic scramble. Embryogenesis is a highly ordered process that results in specific cell types appearing along major spatial axes, and that order is everything. As the zygote divides again and again, the new cells are guided by precise cell division and differentiation, steered by instructive cues and gene regulation. A crucial idea appears here: cell fate is established early in embryogenesis, and as it is established, cells become increasingly restricted in their potential fates. In other words, an early cell keeps many doors open, but step by step those doors begin to close, committing each cell to a particular role in the organism that is taking shape. The very first divisions have a special name: cleavage. Cleavage is the rapid division of the zygote into smaller cells without growth. Read that carefully, because it is the surprising part. The cell number climbs quickly, but the embryo itself does not get bigger. One large cell is simply partitioned into many small ones. These rapid divisions lead to the formation of a blastula, a hollow ball of cells with a fluid-filled cavity. So cleavage takes a single bulky zygote and turns it into a tidy, multicelled sphere with a space inside. This step matters more than it might first appear, because cleavage is crucial for establishing the body plan. The simple-looking ball of cells is already setting the stage for everything that follows. Next, that hollow ball is dramatically rearranged in a process called gastrulation. Gastrulation is the process by which a blastula transforms into a gastrula, and it does so through the movement of cells into a multilayered embryo. This is the moment the embryo stops being a simple sphere and becomes something with structure and depth. Gastrulation accomplishes three things at once. First, it transforms the blastula into a gastrula. Second, it is essential for establishing the body axes, the directions that will define front and back, top and bottom. And third, it establishes the three germ layers. Those three layers are the key product of gastrulation, and they are so important that they deserve a closer look of their own. The three germ layers are formed during gastrulation, and they are named ectoderm, mesoderm, and endoderm. Think of them as three founding populations of cells, each assigned a different part of the body to build. The ectoderm forms the skin and the nervous system, so the boundary you present to the world and the network that lets you sense it both trace back to this single layer. The mesoderm forms the muscles and connective tissue, the parts that move you and hold you together. And the endoderm forms the lining of the gut and respiratory tract, the inner surfaces where you take in food and air. So three layers, laid down in one process, give rise to the full set of tissues and organs. The diversity of the body has a remarkably simple origin. This raises the deepest question in development. If every cell came from the same zygote, every cell carries the same genome. So how can a muscle cell be so different from a nerve cell? The answer is cell differentiation, the process by which cells become specialized and take on specific functions. The trick is that cell differentiation is a result of differential gene expression, where specific genes are activated in different cell types. Same instruction manual, different pages read. Some genes, the housekeeping genes, are expressed in all cells, because every cell needs them. Other genes, the cell-specific genes, are expressed in only certain cell types, and those are the ones that make a muscle cell a muscle cell. Differentiation, then, is not about changing the genes, it is about choosing which genes to switch on. That idea, that switching genes on and off creates different cells, is the engine of development as a whole. Gene expression is a key regulatory mechanism in animal development. It determines cell fate and tissue formation by controlling the production of specific proteins. So the proteins a cell makes decide what that cell becomes, and gene expression decides which proteins get made. This is why understanding gene expression is crucial for understanding the complex processes of development. Notice how this ties the whole story together. The instructive cues we met during embryogenesis, the restriction of cell fate, the differences between cell types, all of it ultimately runs through which genes are being expressed where and when. Development is, at its heart, the controlled reading of a single genome. If cell fate becomes restricted over time, we need a way to describe how much potential a cell still has. That measure is cell potency, the ability of cells to differentiate into various cell types. Potency comes in a clear hierarchy. At the top are totipotent cells, which can produce all cell types of an organism. Just below them are pluripotent cells, which can produce all body cell types but not extraembryonic tissues. Then come multipotent cells, which can produce several cell types. And at the bottom are unipotent cells, which can produce only one cell type. This latter is essentially the restriction of fate made concrete. As development proceeds and cells commit, they slide down this ladder, from many possible fates toward just one. Some cells hold on to this potential, and we call them stem cells. Stem cells are undifferentiated cells that can divide indefinitely and differentiate into various cell types. That combination is what makes them so powerful. They renew themselves, yet they can also become specialized. In animal development, stem cells are crucial for the formation of tissues and organs, supplying the raw material from which structures are built. But their job does not end when the body is finished. Stem cells also contribute to the maintenance and repair of tissues, working throughout the entire life of an organism. So development is not just a phase that happens once and stops. The same kind of cell that helped build you in the embryo keeps quietly rebuilding you, day after day, for as long as you live. We can now name the whole machinery of development. There are four major events: determination, differentiation, morphogenesis, and growth. Determination specifies cell fate. Differentiation leads to cell specialization. Morphogenesis shapes the body. And growth increases the size of the organism. Together these four turn a zygote into a finished organism. But here is the part that surprises many people. These developmental mechanisms are not limited to early development. They are also involved in tissue maintenance, in cancer, in tissue regeneration, and in tissue engineering. Studying them gives insights into cell regulation and tissue formation, and those insights have real applications in regenerative medicine and tissue engineering. The same rules that build an embryo turn out to be at work, for better or worse, throughout biology and medicine. Plants build themselves too, and they follow the same logic with their own twist. Plant embryogenesis establishes the basic body plan along two principal axes, an apical basal axis and a radial axis. Shaping that plant is morphogenesis, which in plants involves cell division, elongation, and differentiation, guided by environmental cues and gene regulation. But plants do something animals do not. Plant growth is driven by meristems, regions of undifferentiated cells that can divide indefinitely. Primary growth is the elongation of stems and roots, driven by apical meristems, while secondary growth is the thickening of stems and branches, driven by lateral meristems. Because of these meristems, a plant can keep growing and forming new parts throughout its life, a strikingly different strategy from the animal body we have been describing. We end by stepping all the way back to see the finished result, the animal body plan. Animal body plans refer to the general structure of an organism, including the arrangement of organ systems and the integrated functioning of its parts. It is the big picture blueprint that all the steps we have followed were quietly working toward. To compare one animal with another, biologists categorize body plans by a handful of features. Body plans are categorized based on symmetry, whether radial or bilateral, on body cavity structure, whether coelomate or acoelomate, on segmentation, on the type of appendages, and on the type of nervous system. So from a single zygote, through cleavage, gastrulation, germ layers, and differentiation, an organism emerges with a definite, classifiable form. That is the journey of development, from one cell to a whole body plan. See you next time.